of snow or what?
Not snow. Yeah, here you know that you are here, right, in California.
Yes,
So
so
no.
there is a vast difference between inside and outside.
So no snow, just normal, like around maybe 10 degrees Celsius
Okay, so it
outside.
doesn't snow there at this moment.
No
Okay,
snow.
okay,
It's a
then that's
very
good.
perfect weather, like a very pleasant weather.
That's good. So one more minute, then we'll start.
Yeah, no problem, no problem. So how is the Zoological Association is now going?
Yes, so I think after the time, lot of other things also. So this is a new addition in our Zoological Association of Burdwan. So you are also a former member, right? So
Yeah, I was a,
that's good
I
that
think
we
I was like from my early days I was the member.
exactly. So yeah,
I
so we
used
used to study there, and we used to sit
Yeah, I used
together,
to
right?
study. I used to buy books. That time, DG sir was there, Dr. Dilip Goel.
Yes, yes.
Uh,
He'll be attending your seminar today also.
okay. So that time,
All right, doctor.
Arindamda.
Yes,
So we
please.
used to go to with Arindamda to buy books from College Street.
Right,
Okay.
right, right.
So it was a very nice experience, and this is kind of a very important part of my life.
Unforgettable moments,
Yeah,
and also very crucial, right?
yeah.
Okay, so Dr. Ghosh, shall we start? And Dr. Mojumdar is also here. Dr. Manavesh Mojumdar sir is also here. Did he join? No.
Could you hear me? Could you hear me, Sham? Could you hear me?
Yes, I do.
Yes, I
Yes, I
joined. This
do.
is the high time to start the seminar. Okay.
Yes,
Already
please.
we are late
Yeah,
of
yeah.
5 hours, 5 minutes. Huh? Okay.
Yes, so
Okay,
okay,
okay.
let us start. So
Sam,
hello everyone. This
I have
morning
a
the
quick.
task of This morning, the task of introducing our guest of honor and speaker is a great privilege for this session organized by the Zoological Association of Burdwan. I feel elated to introduce a speaker who also happened to be my senior in the college. Today, we have Dr. Biraj Mahato, currently serving as an assistant professor in the Children's Hospital, Los Angeles University of Southern California, United States. Dr. Mahato, after passing his bachelor degree from Vivekananda Mahavidyalaya, Burdwan, completed his master's from the University of Burdwan, followed by which he pursued the doctoral degree from the Indian Institute of Chemical Biology, Kolkata. Dr. Mahato spent a couple of years in the various universities in the United States, including the University of Kansas Medical Center, University of Pennsylvania, UNT Health Science Center as a postdoctoral research associate and research scientist. Before joining as an assistant professor, he was a senior scientist in nanoscope technologies, United States for almost a year. Dr. Mahato has authored numerous publications in peer-reviewed journals, which are of international repute with very high standard like Nature. We'd also like to convey a sincere thanks to all the attendees, including the students who are making this session successful. Dr. Mahato will speak on the regulation of transcription in eukaryotes. Let's welcome him and begin with the session. Dr. Mahato, please continue.
Thank you, Sam, for this very good introduction, and I would like to thank you and thank all the members of Zoological Association and like all the students. So I have a quick question. My first question is it good if I proceed with like a language with the English, or you make like English and Bengali is fine? Because why I'm asking, because there are so many students, and I imagine might be many people from Bengali medium. So do you think it is fine?
Please, as you wish, you can continue, please.
Oh, okay. So just so now, can you see my? Now you can see my screen, right? You can see my screen.
Yes, sir.
So, hello, I'm Biraj Mahato. So, I'm an assistant professor from Children's Hospital, Los Angeles, and today I'm going to talk about regulation of transcription, a practical approach. So, regulation of transcription is a very basic subject to study, I think there are many people in Burdwan who can teach you about this, how transcription happens, what are the regulatory elements. They can even know better than me. So here I'm going to talk about how you use this knowledge of regulation of transcription in disease concept, in this disease context. So if you see here that a gene is always controlled by some core elements of the genome, and those are called enhancers, some called silencers, and of course promoters. So those are the places where RNA polymerase and other enhancers, other transcription factors binds, and they make a transcription complex, and that helps for gene expression positively and negatively. So you can see, so these are very, very important elements. Like for enhancer is just a DNA sequence where other activators can bind. Activator is a protein factor. And core promoter. So this is usually a 1,000 base pair upstream of the gene starting point. So these are the basic elements for eukaryotic gene regulation. I'm not going to talk about prokaryotic gene regulation because you can read any books, but I'm just trying to tell you how we apply this knowledge for managing disease and finding out new therapy. So the whole in the whole part, I will cover how the study of gene regulation is useful for regenerative medicine and how gene knockdown will be useful to treat neurodegenerative disorders, and also how you will use RNA sequencing and ATAC sequencing for regenerative cell therapy. And finally, the CRISPR-Cas9 gene editing system to treat retinal neurodegenerative diseases. This may not be included in your subject or maybe in your syllabus, but I can tell you these are the newest things happening in this world, and I think you should know it. It should at least get touch of this. What are these? What are the application of these things like RNA sequencing, ATAC sequencing, CRISPR-Cas9 gene knockdown studies? So these things. So here is one of the figure from one of my paper that has been published in Nature. So this is you can see in the green part. So this is a converted cells called CIPCs. It's called chemically induced photoreceptor cells. And in these cells, how they actually maintain that photoreceptor identity has been described. You can see one of the part is there is the NF-kappa B. So this is a transcription factor, and that transcription factor binds to another gene's regulatory element that just I described, like for the enhancer, maybe in promoter, maybe in silencer, so they can control the expression. So what I found from my research that NF-kappa B is a controller of a gene expression called ASCL1. So that actually NF-kappa B controls expression of ASCL1 that helps photoreceptor identity making, and that helps to generate photoreceptor actually. So here I'm going to talk how you can use this information or our information that we are going to study. So here you can see. So there are two transcription factors here I'm going to talk about. One is HIP1 alpha, that is called hypoxia inducible factor 1 alpha, and I found that HIP1 alpha can increase the expression of this gene. Is called PGC1 alpha. So how you practically use this information? So how you know that really HIP1 alpha is controlling PGC1 alpha? So there is a way called RVIST analysis. You can see in the A. So this is a very like a usable search engine, you can find just Googling. You can just, if you go for Artista, you can see and you can find out a particular gene's regulatory regions. So if you just give a search for this gene and find out regulatory regions for HIP1 alpha, you will find that this is the region, the sequence where actually they help to express this particular gene. So how we can prove that this is a enhancer? So this is the point. That is what I'm trying to explain. So there is a vector that is called PGL3 basic vector. So in this vector, you can clone different genes, different part of the genome, like for enhancers, like for different kind of silencers. So you can see here that this is only PGL3 vector that has a luciferase. So luciferase is a reporter. So when gene will express, it will show you some signal, and so that you can tell that, okay, gene is expressing. And if you add a promoter in upstream of that, you will find some expression of the luciferase. Now, if this enhancer is really helping and upregulating or downregulating the gene expression, if you add them in the upstream, it's just a cloning inside a vector. So you can see in this case, in my case, it was down-regulating. So it was negatively regulated. And if you take out that particular sequence where the particular inhibitor binds, you can see the opposite effect. So this is the practical approach, how you can use your knowledge of enhancer, activator, gene regulation in practical term. So you have to go for a gene cloning with all the promoters along with the luciferase reporter, your enhancers or silencers, and also a mutated part of the enhancer and silencer. And you, after cloning that, you just you can just transfect them into any kind of cells like hex cells, and you can take that cell lysate and you can measure the luciferase expression. So this is how you can see that really a particular sequence acts as an activator or not, acts as a enhancers or not. So this is how actually you can practically or experimentally explain. So this is the particular vector that I have used. So all this, what I'm showing, is just published research from my part. So next is a TET system for gene regulation. It's called TET off and TET on system. So this is a system actually taken from the bacterial system, and in this case there is you can see RTTA. It's called reverse TET transactivator. This is just a protein, and it can bind with a particular sequence. It's called tetracycline response element, TRE, and there is your gene of interest. So there is TET off and TET on system. So what you can do, you can add doxycycline. Doxycycline is kind of antibiotics. So if you add antibiotics, it can bind with this RTTA and it express the gene. It's called on system. And sometimes there is an opposite way. So two systems are available. One is opposite way. If you add doxycycline, the gene will go off, so that the RTTA cannot bind in the tetracycline response element. So this is the whole gene regulation system. You will like read in your textbook. But what scientists do, scientists take out this part, take out these genes, they clone it in the vector, and then they insert it into a mammalian system, into cell culture system, into tissue, so that use this system for expression of the gene or maybe down regulation of the gene at your wish. So for example, how you can apply this? So for example, like if you think that some of the genes is upregulated, like amyloid beta precursor generating genes are upregulated in Alzheimer's disease. So you want to knock down it. So how do you do that? So you can use this. So you can use this particular system to downregulate that particular gene expression, so that it might be helpful for Alzheimer's disease regulation. So this is how the information of gene regulation you can use practically to manage the disease. Here, one of the test system that I have used in my previous research, you can see this is called TET PLK1 Neo. Again, I'm telling you, it may be very new to you, but these are the very basic things, how you apply your knowledge of basic science into a application. That's why I'm telling you, because this might not be very relevant to your subject, but people should know that. So this is called TET PLKO Neo. This is a vector. It's available in a company called AdGene. So you can see here is some TET repressor, some sequences. We can put your particular sequence here. So what I did, I used my SHRNA sequence to downregulate specific gene in my stem cells paper. So how you use that? So after cloning, whatever your gene of interest, maybe a SHRNA. SHRNA is kind of a small, it's called short hairpin RNA. So it actually helps to downregulate a particular gene expression. So you can clone it into an AAV vector, and then you transfect them in the hex cells, and then you make the virus, and then that virus you can actually transfect to the target cells. And if you add doxycycline, that virus will express that particular gene. Whatever gene is, if it is SHRNA, it will express the SHRNA, and you can see the downregulation of your particular gene. So you can think that a system that is present in bacteria that helps for control expression in particular gene can be used in the mammalian system, and that can be useful to control gene expression, and that ultimately useful to manage disease or make new therapy. Here, the next part I'm going to talk about, it's a very new technology that I performed in my Nature paper. It's called ATAC sequencing. It's called Assay for Transposase Accessible Chromatin Sequencing. Why you know that? Because this is also related to gene expression. So in this technology, you can see there is a TN5 transposase element, and that element, it isolated from I think from a different species. I actually forgot which species, but you can clone that and use it in a mammalian system. So think about that. We are using different systems, like from bacteria, from virus, from other kind of different like fish. We are taking those genes, taking those regulatory elements, and inserting it into the mammalian system so that we can achieve that goal. So that is here we are showing. So TN5 system you can use so that it can bind in different mammalian different parts of mammalian genome, specifically in the open chromatin regions. So in the open chromatin regions, it can bind and then cut, and you can use a particular adapter, and then you can sequence that particular genes. So only the open chromatin regions you can see where this TN5 transposase can bind, you can sequence. So what it indicates? It indicates that open chromatin regions are active chromatin regions. That means they are expressing particular genes. So in our study, we wanted to see what are these particular regions, what are those genes where the chromatins are open. So we can find out from our ATAC sequencing that can show that, okay, the genome wide, like we have 33,000 genes, this gene are kind of in the open region, that there is a chance that these genes are expressing. So now how you validate this data? So your control of gene expression here is applicable. So the chromatin is open. That means there are enhancers, your silencers, your activators, all are very accessible. They are going, binding and expressing your genes. And now you validate this data. So the idea is that when you should see in the other experiments that the genes where this particular TN5 transposase can bind, and they can show the expression in the ATAC-seq, that should correspond to expression of particular genes. So this is only showing that, okay, your chromatin is open, and it might be that the chromatin is kind of active and they are working. But how do you prove that? So there is a technology called RNA sequencing. So you can take out the whole RNA from a cell, a single cell. It's called single cell RNA sequencing, and you can go for the sequencing for that. And if you sequence, it can tell you all the particular RNA present in that stage of that cell. So this is one of the results from my photoreceptor cells that we published in Nature. You can see that these are the genes are upregulated. You can see this color. This means you can see the log 2 CPM. So that means it's showing that this is in log scale, and this is a very low expression. And as it goes in different colors, as you go from left to right, it is kind of expressing high. You can see ROR beta. So these are our cells that of interest that we created, and that cells have this kind of expression. But the other cells, those cells we started from, those cells do not have the expression of those particular genes. So it is clear that these genes are upregulated. Now, how you use your regulation of gene expression knowledge here? So that means these genes are open, so their RNA is transcribed. So this particular result should coincide with this ATAC sequencing. That should show that, okay, that particular regions, that particular genes, those are upregulated in your RNA sequencing, are also here in the ATAC sequencing. The chromatin is open. So this is how you can the whole genome, whole transcriptome, and the whole chromatin knowledge you can apply, and all it is related to regulation of gene expression. So these are the very basic. So you always read like a particular gene, how they are regulated. No, we are talking about the whole genome, all the genes, all the chromatin, how they are actually behaving. That is very important. So this is one of another illustration showing how ATAC-seq actually you can present. So these are, you maybe say, oh, what are these? But these are very easy to explain when you put your head. It's very easy. So it's showing that this is a particular like ASCL1 gene. So this gene, this is a particular cell type. This is a particular cell type. This is a particular. I'm not saying all the details about that. But what I'm trying to tell you, you can see in this first map, there is no open chromatin regions here. But if you go to other type of cell, you can see the open chromatin regions here. Chromatins are opening. So with time, these cells changing their chromatin and opening their chromatin so that other activators, other kind of protein factors and promoters are open, and so that they can bind and they can express. So this is a stage 1, this is stage 2, this is stage 3. So clearly shown that stage 1 and stage 2. I'm just making it very simple. So stage 1 and stage 2 has a different chromatin regions. So stage 2 has open chromatin regions. So that means this particular gene should express and should show up in the RNA sequencing data. In the total RNA, so it should express. You can see here, it is upregulated. It is upregulated here. It is less, it is high. So the same sample. So this is a kind of regulation of your knowledge apply to different kind of next generation sequencing study. And you can see like here, particularly this part, like there is called promoter region, proximal promoter region, you can see here. So here in this cell type, it was 10%. When the cells is converted to other cell type, it is going to 17.6%. So in total, there was a 10% proximal promoter regions were open. But when the cells are converted to this kind of cells, like here RID8, you can see it's opening up more chromatins. So we are talking about eukaryotic gene regulation in global basis, not a single gene regulation. We are talking about how the whole 33,000 genes and maybe other potential genes are regulated by opening chromatins and by synthesizing RNA. So here there is another new technology. It's called CRISPR-Cas9 gene editing system. I think people should know that these are kind of these days, these are like very basic things that going on in the laboratory. So CRISPR-Cas9 is also a system that actually hijacked from the bacterial system. So this is also a bacterial immune system against the virus. So there is a CRISPR sequences where a Cas9 particular protein can bind. So what happens when virus actually affected the bacteria, specifically the bacteriophage, affect the bacteria, what happens? It keeps its particular genome sequences inside the bacterial genome. And that is called like a kind of, that is the palindromic sequences, where those particular Cas9 protein can bind, and then they help in bacterial survival. So you can find a lot of information about that. Just Google search. What I'm going to tell you that how a particular gene regulation system that is helpful for bacterial defense against virus is using or useful in mammalian system. Here you can see that this is what currently I'm doing in my lab, that we are using CRISPR Activator. So what is our goal is to activate particular gene in a particular cell type so that we can convert a particular cell to other cell type. This is called cellular reprogramming. So what we are doing, we are taking skin cells and we are trying to convert them into eye cells, eye retinal cells, retinal neurons. So what we are using a DCAS9 system that is upregulated, that helps to upregulate a particular gene, particular transcription factors in the skin cells that help convert those skin cells to eye cells. So here you can see that there is a guide RNA is necessary that actually guides the DCAS9 to bind to a particular genome. So guide RNA you can. So I'm not going to going in details about that. You will find a lot of information on this, and then just Google. What I can tell you that guide RNA binds with the DCAS9 system. So you can clone the whole system in a particular vector and then use them in a particular, like in our skin cells we are using. So we first clone this DCAS9 system, guide RNA sequence in a particular vector that can produce AAV, it's called adeno-associated virus, and that infect that virus into particular cell so that they can express these particular system and activates particular gene. So this is called CRISPR Activator that we are currently using. And recently, one of the paper has published this, and I'm currently using this in my lab as well. So the vector is contains the Cas9, and you can see there are two guide RNA 1 and guide RNA 2. So all these are genes. You are cloning in a particular vector. That's it. And then you are using a particular promoter and then cDNA sequence. The whole sequence, you make a virus. You can see in the E. So that virus you are injecting into the eye. So idea is you want to express this particular gene as well as you want to destroy the bad gene. So in the retinitis pigmentosa, this is a very common form of blindness in all over the world, and millions of people are affected. What happens? There is a mutation in the RP gene. So what I'm currently doing, I'm using this method to destroy that particular bad genes and then overexpress the good gene for corresponding to that. So that the bad gene is just go out, and then the actual gene is overexpressing in the eye so that people can restore their vision. Those cannot see due to that particular mutation. So this is what. So we clone this whole system, make AAV virus, and thus inject into the blind mice. Those have that particular mutation, and see what happens in our following experiments. So this is the final video. So whatever you are doing, like you are making cells, you are making virus, all using your knowledge of regulation of gene expression, and then how you are applying. So in this particular eye, this is a disease eye. In this eye, this is called due to this happens due to this disease eye is kind of a AMD. It's called age-related macular degeneration, where a particular retinal cell type called photoreceptors are degenerated, and due to that people cannot see, they are blind. So what you can do, you can use your knowledge of gene regulation and make those viruses. You can make those cells and cells transfected with viruses and injected into the eye, so that your cells or your virus will go into the eye. So this is the retina, okay? And now you can see it's an example for cells, and the cells will go there. Cells will, so you can see the other cells are died already. So the new cells are now growing up, so that these cells now function, and it's functionally replaced the lost cells. So this is the application, how you can use your knowledge of gene regulation in real disease context. Thank you very much for listening my talk. And this is my institute. I'm in the 4th floor. My lab is in the 4th floor. And thank you very much for your attention. And I would love to answer any questions you have. Thank you.
Thank you, Dr. Mahato. Students are requested to ask questions if they have any.
Yeah. Any question? Those you are thinking that English is a problem, you can ask me in Bengali, no problem. Any basic question you have? Not only that, what I talked about.
I think, so it's a very interesting piece of study. Can I ask you a question?
Yeah, sure.
Yes, so is it, so the same technology, is it possible to apply for other organs or other kind of systems apart from retinal cells?
Yeah, people are using already in the in liver, different kind of liver diseases, chronic liver diseases they are using. So people are using in the heart diseases as well. So for the beauty in the eye is that eye is immune privileged. So if you inject something, there is no immune response. That's the beauty. So that's why anything you can try in the eye. So, but if you go for the liver, that there might be some immune response. So that's why before you apply really into the liver or maybe other organ, you have to consider that particular part of immune regulation, or maybe generating immunity against that particular. Yeah.
Thank you. And how? Yes, please go ahead.
Dr. Mahato, may I ask a question? That is, do we utilize this technology against P10 genes?
Which genes?
P10 genes responsible for cancer. There is a mutation.
Yeah, definitely you can use. But the problem is that in cancer, this is a multi-factor. Like here, this is monogenic. Like for a particular.
So targeting a particular gene cannot be
very easier. But
in
cancer, it's not only like gene mutation; it also other type of.
So many mutations there.
Not only mutations like phosphorylation. You cannot correct phosphorylation, like acetylation. You cannot correct that. So something but it is very difficult to manage that kind of disease. But a monogenic disease, like a particular gene in a single gene, there is a single mutation, you can control.
Sir, I have a question.
Yes, sure.
Sir, can we create brain cells or neurons to treat Alzheimer's disease? For what you have said.
Yes, you can do that. I do that in my lab. Yeah, it's possible. So what do? We in our lab we took skin cells, and then we overexpress 4 transcription factors that actually helps to convert them to neurons. Here is the regulation of gene expression. So you're overexpressing particular genes. How you overexpress? Here is all the knowledge of enhancer, all the knowledge of promoter will come, right? So when you overexpress those 4 particular genes in the skin cells that will convert to neurons for Alzheimer's disease, you have to have your knowledge about promoter, your knowledge about enhancer, your knowledge about silencer, how you clone them. So the whole thing. So this is a very good question for related to regulation of gene expression. Yes, it's possible.
Okay, sir. Thank you.
So how much, I think, how far you think that you are to reach the technology from the bench side to the bedside?
So for the technology that I published last year, that one of the company called CIRC Biosciences, from this is a Chicago-based company. So I think you can go online and you can check. So that company actually kind of take the license. They took the license from us. So I'm the kind of one of the inventor in the patent. So they are kind of what they are doing. They are recruiting now patients for retinitis pigmentosa, for maybe for AMD. So they are collecting the skins and making the skin cell bank. And what their plan is, in each case, so they will convert them to our neurons, that eye neurons, that helps to kind of restore vision. So and then they'll inject them back to the same patient. So we actually applied to FDA, Food and Drug Administration, US government, and we got I think it's called some drugs. There is some kind of drug status. It's called I forgot the name exactly. It's I cannot remember. So there are different stages. One is a stage is called you have to take that drug status first, and then you have to recruit patients. So you can go for that particular website, CIRC Biosciences. So you can find out. Oh, it's an orphan drug status. So if you get orphan drug status, you can recruit the patients. And now we will submit the whole protocol to FDA in March, and then FDA will say, okay, you can go move forward. So there are reviewers; they will review that, and then we can apply it to the human. And whether in human it will work or not, that we don't know right now. Yeah. So this is how it's like from bench to bedside is actually move on. So what I'm telling, trying to tell all the students that look, your particular knowledge of regulation of gene expression is applying to make a particular technology, and that is going to actually treat some diseases. Here we are using AMD, called age-related macular degeneration, where eye neurons are degenerated. So it's not like you always read like, okay, there is a promoter, then that is not that is not very interesting to you. But when you think about the application, the whole thing, then it will be very, very interesting. Yes.
Is there any more question? No? Okay. Since there are no more questions, so I would like to request Dr. Banamesh Majumdar on behalf of Zoological Association of Burdwan to express the vote of thanks.
Thanks, Shyam. I am on behalf of the Zoological Association. I thank Dr. Biraj Mahato for his nice deliberation upon today's topic with all sorts of global informations right now available. It is also a matter that a lot of thanks goes to the students who have joined this session to enrich their knowledge level to the best of this topic. Thanks to all the active personage of the ZAB in making this program a success. Last but not least, I thank the Google Meet platform for smooth and proper convection of this seminar. With this few words, I conclude the session. Again, thanks a lot.
Thank you very much. This is a kind of very emotional. I am basically emotionally attached with this organization. So I just want to tell you something about my experience with Zoological Association to our students. So I think Sam, I have that. I would have that opportunity, right? Can I tell
yes,
them?
Yes, please.
Yes. So keep in mind that this Zoological Association plays a very great role in my journey. When I was a BSc student, I used to go there there's very good different books, and I used to buy books with, I told you that I used to go for buying books with Orindamda, and I would like to sincerely I would like to tell my thanks to Dr. Dilip Guha. So he was one of my teacher and one of our like great teacher in my life. So he actually give us all this motivation. So go there, buy new books, and that will give you knowledge. So I just followed that. You can follow that. So if you really believe something that okay, you want to do something, uh, you have that resource there. You can use that. That's a big resource. I don't know how it is right now, but that time it was just a like I think there are 2 floors, first floor and second floor. And second floor, there are a lot of books. We used to go there. We used to read a lot of books. So that was a great experience. And I sincerely like thank to all my teachers and all the Burdwan Zoological Society. So it's very, very helpful. So you can take that opportunity.
Thank you, Dr. Mahato. We are delighted to know the important role that the Zoological Association of Burdwan played in your life to shaping up your career. So with this, we conclude the session. Thank you very much.
Thank you very much. Thank you very much.
Thank you, sir, for your advice.